Okay, so good morning. First of all, I would like to thank the organizers, especially Duncan, for the kind invitation. It's really a privilege to be here. And yes, I'm going to talk about KB 1.5 channels in the pulmonary circulation. This is an area in which our group has been working for many years. During my first part of the talk, I will give a brief overview of the physiological and pathological roles of these channels in the pulmonary vasculature. And then in the second part of the talk, I will share some results we have obtained targeting these channels in the context of pulmonary arterial hypertension. Well, KB 1.5 channels belong to the voltage-gated potassium channels, which together with other potassium channel families like the two podomain potassium channels, the calcium-dependent potassium channels, inward rectifiers of KTP, are functionally expressed in the pulmonary vasculature. I'm going to focus on the role of these KB channels, in particular KB 1.5 channels, in the smooth muscle cells. And surely the most relevant role or best well-known role of these channels is controlling pulmonary vascular tone. So essentially, when these channels are open, the outflow of potassium ions leads the membrane potential more negative, and this precludes the calcium entry through voltage-gated calcium channels, leading to pulmonary vasodilation. And in contrast, when these channels are closed or their activity is attenuated, this results in a depolarization, enhancing calcium entry and promoting vasoconstriction, as well as proliferation, migration, etc. But before moving forward, I think it is important to mention that there are important differences in the pulmonary circulation as compared with the systemic circulation. Now, the pulmonary circulation is a low pressure and a high flow system. You see that the pulmonary vascular resistance is about 1/7 to that found in the systemic vessels. So that it is conceivable that the mechanisms controlling pulmonary vascular tone may differ substantially from those that control vascular tone in other systemic vessels. And a striking example is KB channels, because these potassium channels, the activity of these channels is much higher in pulmonary vascular smooth muscle cells compared to that found or recorded in smooth muscle cells from systemic vessels. So, and why KB 1.5 channels in particular? Well, here you can see an original recording of the KB channel current activity in rat pulmonary artery smooth muscle cells. And when we apply this compound, which is a KB 1.5 channel inhibitor, TPO, you see that the current is markedly blunted, okay? So this strongly suggests that KB 1.5 channels are the main contributors to the total KB current activity, KB currents present in the pulmonary artery smooth muscle cells. Now, what do we know about these channels? We know that these channels are targeted by different factors, different vasoactive factors, and especially by pulmonary vasoconstrictors. So our group and others have found that different vasoconstrictors which have a really relevant role in the pulmonary circulation, like endothelin-1 or thromboxane 2 or serotonin, have been shown to be target these KB 1.5 channels, to inhibit these channels, and this contributes to the vasoconstriction induced by these vasoactive factors. But also another important issue of these channels is that these channels are oxygen sensitive. A long time ago, several groups like Stephen Archer, Kenneth Ware, Jason Joan, they were the first to show that hypoxia inhibited these KB currents in the pulmonary vasculature, and this contributed to the so-called hypoxic pulmonary vasoconstriction, or HPV, which is a physiological response of the small pulmonary arteries which constrict in response to low oxygen levels. And this is very important because that allows to divert the blood to the better oxygenated areas of the lung. So in other words, this HPV response allows the matching of ventilation and perfusion and gas exchange. So our data also confirm that hypoxia indeed inhibited KB channels, particularly KB1.5 channels. Now the elucidation of the mechanisms inducing this inhibition was quite, or create a big debate, a controversy between different authors, saying that this could involve increase or decrease reactive oxygen species. At least in our hands, we found a complex signaling pathway involving an increase in reactive oxygen species. We found that there was an initial increase in the mitochondrial reactive oxygen species, which was followed by a neutral sphingomyelinase-derived ceramide production, which further amplified a reactive oxygen species increment through the activation of the NADPH oxidase, leading to the inhibition of this current, membrane depolarization, and finally the hypoxic-induced vasoconstriction. But beyond and following this suggestion, we found that several of the mediators like ceramide or hydrogen peroxide mimic the effect of hypoxia inhibiting these KB1.5 channels. But beyond the exact mechanisms inducing the inhibition of KB1.5 channels, we have a lot of evidence in favor of the role of these channels to participate in this important response in the pulmonary vasculature as the hypoxipulmonary vasoconstriction. And here you can see how this hypoxipulmonary vasoconstriction is impaired in animals lacking of KB1.5 channel. So in summary here, I've gone through the physiological role of KB1.5 channels in the pulmonary vasculature, but what about the role in the pathophysiology of the pulmonary vasculature in pulmonary vascular diseases? Well, during the last, I would say, decade or probably 2 decades, we have learned that ion channels, and particularly potassium channels, and you see here KB channels, are targeted by many pathophysiological factors involved in pulmonary vascular diseases. I don't have time to go through all the details of these different factors, and they have been excellently addressed in different recent review articles that you can take a look at. But I'm going to move now to the pathophysiological role of KB1.5 channels, and particularly, we've been particularly interested in the role in pulmonary hypertension, and more particularly in pulmonary arterial hypertension. This is a rare but very severe disease that affects the pulmonary arteries. And in these patients, the small pulmonary arteries have a persistent vasoconstriction, and there's also a remodeling of these small arteries, leading to, which is characterized by a medial thickening and muscularization and intimal occlusion, which limits the vascular diameter. And the consequences of this is a progressive increase in pulmonary vascular resistance, an increment in pulmonary arterial pressure, and unfortunately, in many cases, premature death due to right ventricle heart failure. So the pathophysiology of this disease is very complex. We know that the gene predisposition, together with many environmental factors, may exacerbate or inhibit a number of signaling pathways to promote some pathological processes, including inflammation, endothelial dysfunction, and ionic remodeling, which involves essentially dysregulation of potassium channels in different cells present in the pulmonary vasculature. Especially, we've been focusing on the pulmonary artery smooth muscle cells. Now, why KB1.5 channels in particular? Well, there's some potassium channels that have been known to be affected, to be dysregulated in pulmonary arterial hypertension, but KB1.5 channel was the first ion channel to be linked to this disease. This was in the late 90s, where Jason Johns and colleagues reported that KB1.5 channel activity and expression was downregulated in pulmonary artery smooth muscle cells from PAH patients, from patients with pulmonary arterial hypertension. This group, years later, also reported some genetic variations in the gene encoding for KB1.5 channels in the KCNA5 in patients with idiopathic pulmonary arterial hypertension. And more recently, we have analyzed the functional consequences of these variants in the gene. Okay, in particular, we have looked at different variants found in a Spanish cohort of PAH patients. And here are the data we obtained with the 7 variants that we analyzed. As you can see, in most of them, there are no changes in the current amplitude, in principle, no changes in the activity of the channels, but two of these variants were associated with a marked loss of Kv1.5 channel function. And more importantly, these mutations were associated with a decrease of pulmonary artery smooth muscle cell apoptosis. In other words, these mutations increase the viability of these smooth muscle cells, which could contribute to this remodeling that I just mentioned in a few slides before. So these data support the notion that Kv1.5 channel impairment may contribute to the genetic forms of pulmonary arterial hypertension by leading to a more depolarized membrane potential, increasing vasoconstriction and remodeling. Now, what about disease regarding with the genetic forms of the disease? But what about the non-genetic forms of pulmonary arterial hypertension? Well, we have a vast evidence showing a consistent downregulation or impairment of Kv1.5 channels in different models or tissues from PH patients or experimental models. But to sum up, I want to show you this transcriptomic analysis that our group led by my colleague Francisco Pérez Vizcaíno did a few years ago. This volcano plot represents all those genes that whose expression is impaired in pulmonary arterial hypertension. These are lungs from pulmonary arterial hypertension patients. And as you can see in blue dots, you see that many of the genes that are impaired in pulmonary arterial hypertension can be either downregulated or upregulated. Okay, the amount is quite similar. But if we focus on those genes encoding for cation channels, we see that the majority are downregulated. The majority that are impaired in PAH, okay? And especially potassium channels, and including Kv1.5 channel and TAS1 channel, the other potassium channel that has been one of the other potassium channels that have been involved in pulmonary arterial hypertension. But what is really interesting is that this downregulation of Kv1.5 channels found in all the patients is also found in different forms of the disease, like the idiopathic pulmonary arterial hypertension, as well as in pulmonary arterial hypertension associated with other diseases like congenital heart disease or connective tissue diseases. So this is a good example of how this Kv1.5 channel seems to be downregulated in general in the different forms of pulmonary hypertension. Now, what about in the experimental models that we use to study this disease? Well, this is a typical, this is probably currently is the gold standard model for studying pulmonary arterial hypertension that involves the administration of this drug. Sugin is a vascular endothelial growth factor receptor antagonist, a single injection, and then exposure to the animals to hypoxia for at least 3 weeks. And this is what happens. This is the pulmonary vascular pulmonary arterial pressure. This is in a normal animal, on an animal exposed to normoxia, and you can see how animals exposed to this model experiment a really marked increase in pulmonary arterial pressure. Okay. Now, if we analyze the Kv currents in cells isolated from these animals, we see that in fact the Kv current is markedly affected, markedly downregulated. And this is observed not only in this model. Well, this is associated with a marked downregulation or inhibition of the DPO-sensitive current, so it's the Kv1.5 current in principle, and also is associated with downregulation of Kv1.5 channel expression. And the same is observed in other more classic models of pulmonary hypertension, like the chronic hypoxic exposure or the monocrotaline-induced pulmonary hypertension. So in general, Kv1.5 downregulation is a common hallmark of the disease in both patients, both genetic and non-genetic forms of the disease, as well as in the experimental models. And therefore, searching for strategies in order to try to restore this affected this affected channel is of great interest for a group and others. So I'm going to show you some strategies that have been tested, like gene therapy with the KCNA5 gene, which was shown to be able to increase the affected Kv1.5 channel downregulation in the chronic hypoxic model, and this was associated with attenuation of the pulmonary vascular resistance in these animals. And more recently, our group and others have targeted microRNAs. In our study, we identify MIR1 as a microRNA that could directly affect the expression of Kv1.5 channels, and the antagomir was able to partly restore the attenuated activity in the hypoxia in vitro model. And a similar approach was done by the Jason Josh group. They found that MIR29B inhibit this Kv1.5 channel activity in cultured human pulmonary artery smooth muscle cells, and more importantly, was able to restore the affected channel activity in a human pulmonary artery smooth muscle from PAH patients. But of course, this will take some time to translate into the clinic. So therefore, we have also trying to look for more feasible strategies, okay, to restore the affected Kv1.5 channel activity, and we have focused on this target, the sigma-1 receptor, that probably many of you are familiar with. The sigma-1 receptor is expressed in the endoplasmic reticulum and may affect the function and trafficking of different proteins, including several ion channels. I have no time to go through it, but so we first did in silico analysis, which predicted a potential interaction of this chaperone with the Kv1.5 channel, and this was confirmed by immunoprecipitation assays. So then we started to examine the effects of different modulators of the sigma-1 receptor. And I'm going to focus on the effects we obtain with an agonist of this sigma-1 receptor called the pre-084. This activator of sigma-1 receptor was shown to enhance Kv1.5 channel activity in heterologous systems, in overexpression systems. But what is more relevant for us, it also greatly enhanced the Kv1.5 channel activity, the DPO sensitive current, in freshly isolated pulmonary artery smooth muscle cells. And in addition, this drug was able to attenuate the vasoconstriction, the pulmonary vasoconstriction, and the pulmonary artery smooth muscle proliferation. So these effects were in total were very promising in the context of pulmonary hypertension, as it was reducing vasoconstriction and reducing proliferation. So that led us to try to test this drug in an in vivo model of pulmonary hypertension, the one that I just mentioned, that involves the treatment with Sugen plus the 3 weeks exposure to hypoxia. Let me tell you that after this 3 weeks exposure with hypoxia, and after treated with Sugen, the animals, the disease is irreversible. So that even when the animals are turned back to normoxia, the pulmonary arterial hypertension persists. Okay, so now that's why one of the reasons this is currently the gold standard experimental model. So we had 3 groups: animals exposed to normoxia, to the pH model, and also treated with the sigma-1 receptor agonist for the last 2 weeks. This is a quite challenging model because, I mean, it's not the same using as a preventive model than as a curative model, because here the pH is already established and the vessels are already remodeling. So what do we found? Okay, so here you can see how, as expected in the PAH animals, the Kv current was markedly impaired. Okay, while treatment with the sigma-1 receptor agonist was able to substantially restore this affected channel. This was here you can see the IV relationships, and also it was the drug was able to restore the resting membrane potential to normal to those found in animals exposed to normoxia. Interestingly, the treatment with this drug also reduced because here you can see that the capacitance was enhanced. This is associated with a hypertrophy of the smooth muscle cells, and this is also partially attenuated. We think that this is because the drug stimulates the trafficking of the drug, of the channels to the membrane. I'm going to go through it quickly because I've seen David is just standing up. So the drug was able to partly reduce the hypercontractility, the vascular remodeling, also the hemodynamics, and the right ventricle remodeling. And during I will just 2 minutes to go through additional data because sigma-1 receptor has been shown to be also modulated by some already commercially available drugs like fluoxetine or fluvoxamine, antidepressants, dimemorphan, and others. So we've been testing also the effects of these drugs on Kv1 channels. Here you can see that fluoxetine and dimemorphan are also able to markedly enhance the Kv1.5 channel activity. This effect is prevented by sigma-1 receptor antagonists, the BD compound, and this effect is even higher in smooth muscle cells from the PH animals, okay? So this is very, very promising. And finally, we have also confirmed the ability of, in this case, dimemorphan, to greatly enhance Kv1.5 channel activity in freshly isolated human pulmonary artery smooth muscle cells. So to sum up, Kv1.5 channels play an essential role in the pulmonary circulation. Their dysfunction represents a hallmark in pulmonary hypertension, so enhancing their activity may represent an attractive therapeutic strategy. I have gone through one potential strategy that is targeting the sigma-1 receptors, and future studies are also needed to know the role of these channels in other cells in the pulmonary circulation apart from the pulmonary artery smooth muscle cells. And I would like to thank all the people from my lab, my collaborators, and my funding agencies. And I would like to thank you all for your attention as well. Sorry.
Nice talk. I was thinking about the MIR1 story, and it's interesting. So it's directly targeting the Kv1.5.
Yeah. It's directly targeting KCNA5. Yeah, I couldn't include that data because otherwise it was a long. Yeah.
Yeah. So it's a direct target. So it reduces the expression of the channel. Right. That is actually what's happening. Yeah. Okay. Thank you.
Thank you.
Question here. Hi, thanks for this really interesting talk. I'm interested in the genetic variants you have showed. I think there were 7, and you showed that 2 of them decreased the current amplitude.
Right.
I guess it was in a heterologous expression system. So did you also check for other mechanisms that could impact the activity, like different parameters, voltage dependence, or kinetics?
Yes, we did. I just showed a summary, like only the total current at plus 40 millivolts, I think. That we didn't really see that much difference in many electrophysiological characteristics. So we think that essentially, especially in the variant that caused the most loss of function, there was also a decrease in the expression. So it affected the trafficking more than I think there was a very small effect on the kinetics, but it was not that relevant. Okay, and we didn't study the effects of regulatory subunits and how that could affect as well. So it was just Kv1.5 with a different. So the alpha subunit with the different variants.
Exactly, because I also thought another mechanism could be like the lack of protein interactions in a native environment, because some of them were on the, I think, extracellular loops. So maybe in the native environment, they would have an.
Sure, I mean, and the ones that we didn't see an effect, we cannot rule out that they could also be impaired in, by different proteins, regulatory subunits, whatever, that are in the pulmonary artery smooth muscle cell because they were overexpression systems. You're right.
Thank you.
Okay, so in the interest of time, I'd like to thank our first three speakers.